This video is on cells. Start with the prokaryotes and eukaryotes. Eukaryotes have internal membranes while prokaryotes do not. A prokaryote is a unicellular organism that lacks a membrane-bound nucleus, mitochondria, or any other membrane-bound organelle. Prokaryotes are divided into two domains, archaea and bacteria. The first living organisms were prokaryotes. A eukaryote is any organism whose cells have a cell nucleus and other organelles enclosed within membranes. Eukaryotes can be unicellular or multicellular organisms. The defining feature that sets eukaryotic cells apart from prokaryotic cells is that they have membrane-bound organelles, especially the nucleus. Eukaryotes evolved about 1 billion years ago, while prokaryotes evolved 3.6 billion years ago. Oh, there we go, a little closer. Sorry about that. The nucleus contains most of the cell's genetic material, organized as DNA molecules combined with a variety of proteins to form chromosomes. The genes within these chromosomes are the cell's nuclear genome and are structured in such a way as to promote cell function. The nucleus maintains the integrity of genes and controls activities of the cell by regulating gene expression. Ribosomes are both synthesized and produced in the nucleolus. The ribosome is the site of protein synthesis. Ribosomes link amino acids together in the order specified by messenger RNA molecules. Ribosomes consist of two major components: the small ribosomal subunit, which reads the RNA, and the large subunit, which joins amino acids to form a polypeptide chain. Each subunit is composed of one or more ribosomal molecules and a variety of ribosomal proteins. The Golgi apparatus receives proteins and lipids from the rough endoplasmic reticulum. It modifies some of them and sorts, concentrates, and packs them into sealed droplets called vesicles. The Golgi apparatus is located near the nucleus and has several flattened membranous sacs. The smooth endoplasmic reticulum is devoted almost exclusively to the manufacture of lipids, and in some cases to the metabolism of them and associated products. In liver cells, for example, smooth ER enables glycogen that is stored as granules on the external surface of smooth ER to be broken down to glucose. Smooth ER is also involved in the production of steroid hormones in the adrenal cortex and endocrine glands. Small amounts of smooth ER are found in liver cells, where one of its main functions is to detoxify products of natural metabolism and to detoxify overloads of ethanol derived from excess alcoholic drinking and also barbiturates from drug overdose. To assist with this, smooth ER can double its surface area within a few days, returning to its normal size when the assault has subsided. Rough ER gets its name because it is studded on its outer surface with ribosomes. These are called membrane-bound ribosomes and are firmly attached to the outer cytosolic side of the ER. Rough ER is found throughout the cell, but the density is higher near the nucleus and the Golgi apparatus. Mitochondria are rod-shaped organelles that can be considered the power generators of the cell, converting oxygen and nutrients into adenosine triphosphate, which is ATP. ATP is the chemical energy currency of the cell that powers the cell's metabolic activities. The process is called aerobic respiration and is the reason animals breathe oxygen. The main role of chloroplasts is to conduct photosynthesis, where the photosynthetic pigment chlorophyll captures the energy from sunlight and converts it and stores it in the energy storage molecules ATP and NADPH while freeing oxygen from water. The mitochondria produce ATP during aerobic respiration. You should know that chloroplasts produce ATP for photosynthesis in plants, while mitochondria produce ATP for aerobic cellular respiration in animals. Lysosomes break down cellular waste products, fats, carbohydrates, proteins, and other macromolecules into simple compounds, which are then transferred back into the cytoplasm as new cell-building materials. To accomplish the tasks associated with digestion, the lysosomes utilize about 40 different types of hydrolytic enzymes, all of which are manufactured in the endoplasmic reticulum and modified in the Golgi apparatus. In animal cells, the plasma membrane is the only barrier separating cell contents from the environment, but in plants and bacteria, the plasma membrane is also surrounded by a rigid cell wall. Bacterial cell walls are composed of peptidoglycan. Yeast cell walls are composed of two layers of beta-glucan, the inner layer being insoluble to alkaline conditions. Both of these are surrounded by an outer glycoprotein layer rich in carbohydrate mannan. Plant cell walls consist of multiple layers of cellulose. These types of extracellular barriers confer shape and rigidity to the cells. All cells are surrounded by a plasma membrane. The membrane is composed of a phospholipid bilayer with proteins dispersed throughout the layer. The membrane is also covered in places with cholesterol molecules and proteins. The plasma membrane is selectively permeable and regulates which molecules are allowed to enter and exit the cell. The cell wall gives the plant its actual shape. It acts as a gatekeeper because it determines what can come in and out of the cell in order to keep the cell protected. The cell wall protects the plant and cells from the many insects and pathogens that could harm the plant. There are holes all over the cell wall called plasmodesmata. These are holes that allow for nutrients to enter the cell as well as waste to exit the cell.